Bismillahirrahmanirrahim. Welcome back, friends. Welcome to another video lecture by Kami Biology. In this video lecture, we are going to talk about allylamines. So these are actually the videos lecture regarding with the antifungal drugs. So what we are going to talk about? We are going to talk about allylamines. So first of all, what is allylamine? Allylamine is the antifungal drugs which are specially used for the means which are specially used to kill dermatophytes, or we can say aspergillus species, or we can say candida species. We can say that allylamine are those drugs which can also stop or which can inhibit the squalene monooxygenase enzymes, which are specially responsible to convert the squalene into lanssterol, and then lanssterol will passes from the cytochrome P40 alpha demethylase, just like that enzyme. I forget the correct name of the enzyme. And after that, once the lanssterol passes from this enzyme, then we get the gasterol, and after the gasterol, then it will be added to the cell membrane, by which the cell membrane are flexible and we can say that rigid. So all these are the steroid biosynthesis. So we can also say that these drugs can actually stop or inhibit the synthesis of the steroid. So how? Inshallah, it will be explained later. First of all, let's talk about the special drugs which are used in allylamine. These kinds of the drugs, let me write the name. So the first drugs are niftyfine. The second drugs in this case are the terbinafine. Flunarizine, butinafine. Butinafine. So these are the special drugs which are used in allylamines. So how this allylamine will look like? If we see the structure of the allylamine, so I'm not drawing the correct structure, but the allylamine will look like in this form, some like that, and here is nitrogen group. So this is the diagram of the allylamine. So these are the allylamine, means allylamine will look like in this form. Now, before going to explain the mechanism of action of allylamine, first of all we need to understand that how actually the steroid of the fungi are synthesized, means that gasterol are synthesized, or we can say what are the squalene and what are the squalene monooxygenase enzyme, what are the main function of squalene monooxygenase, all we talk about there. First, once we get the idea about that, then the mechanism of action will be easy. So what actually happen inside the fungi cell? We have a molecules just like that. This is not the correct structure of the squalene, but we have a molecule just like that. We simply call that what? As I draw with a black marker, we call that squalene molecules. Squalene molecules. Oh, these are the squalene. What actually happened? They are passes from the enzyme. They are passes from the enzyme. We simply call that enzyme. Let's suppose this is the enzyme. So this squalene molecules will be passes from the enzyme. We simply call it squalene monooxygenase. Squalene monooxygenase enzyme. Once they passes from the squalene monooxygenase, then we get what? We get the lanssterols. These are what? These are the lanssterol. This lanssterol will further passes from another enzyme. We simply call that cytochrome P450. Now this is what? This is actually cytochrome P450. cytochrome P450 alpha demethylase enzymes. So once they passes from here, then finally what we get? We get the agasterols, something like that. These are what? These are the agasterol. This is not the correct spelling. I forgot the correct spelling of the agasterol. Now once then after that they will passes from the agasterol. Once this agasterol, once we get the agasterol, then this agasterol will be added to the cell membrane of the fungi, by which the fungi cell membrane are rigid, strong and flexible. So all these are what? All these are the biosynthesis of the steroids. These are what? These are the actually biosynthesis of steroids. Means agasterol. Agasterol. Gasterol. So that's all about the biosynthesis of the agasterol. Now what are the main mechanism of action of this allylamines? So if we see, they can easily bind. They can easily bind here. So let's suppose these are the structure. So they can easily bind at that area. Or we can say it is actually non-competitive inhibitor, or we can say it is non-competitive. What actually happens once they bind to that area? Then the squalene will be not passes from here, because they can or we can say that actually these drugs can connect it, or they can stop the active site, or we can say they can actually bind to the active site of the enzyme. Once they bind to the active site of the enzyme, then the squalene molecules will be not passes. So when the squalene molecules are not passes, so then the rest of the process will be not occur. Means there will be no lancestrol occur, and there will be no agasterol will synthesized. So when the lancestrol and agasterol are not synthesized, then we can say that cell membrane will be not very strong, and we can say that cell membrane will be very weak, and they cannot they cannot we can say that they cannot bear the osmotic pressure. So once they cannot bear the osmotic pressure, then the cell will be burst, or we can say cell will be lysed. So that's all about the main function, or we can say mechanism of action. So we can say it is actually fungicidal in nature. What? Fungicidal. Why? Because they can kill the fungi. Now let's talk about the antifungal spectrum. So on what kind of we can say that on what kind of fungi they can inhibit the fungi? We can say antifungal. Let me write here, because I don't have any kind of space. Let me write here. So against dermatophytes, let me write. So they can show good activity against dermatophytes and Candida species. Moreover, they can show good activity against the Aspergillus species. And we can also say they can also show good activity against yeast. So these are the fungi which are affected when we give this allylamine. The infection which are caused by this fungi, so they will be treated by this allylamines. And so that's all about the mechanism of action of the allylamines. Now let's talk about the resistance. What actually happened if we talked about the resistance against the allylamine? So resistance are not usually occur in case of the allylamine. Why? Because allylamine is a very effective drug. Or we can say that resistance against the allylamine is not understandable. In some books, they can say that we can say that the enzymes will be mutated, or the genes which are actually present inside the enzymes will be mutated. Once the mutation are occur, it is not occur. It means this process is not occur usually, but sometime this process will be occur. So once the genes inside the squalene monooxygenase enzyme are changed, so we can say then the drugs will be not binded, but it will not occur. And in some cases, then what actually happen, this drugs will be not binded. Once the drug are not binded, so then again the we can say again the biosynthesis of the sterols will be happen. So this is the resistance, but the resistance will be not happen sometimes. It is very effective drugs. So this is a little bit resistance. Then what are the main side effects which are caused by given by this allylamines? The main side effects of the allylamines, let me write side effects. So the side effect which are occur by giving allylamines, these are the GIT disturbance, nausea, headache, itchy skin, or we can say skin itchy. Itchy means rashes on the skin. So that's all about the side effects of the allylamines. I hope you understand about the allylamines, the mechanism of action, and biosynthesis of the steroids, structure of the allylamines, antifungal spectrum, and the side effect of the allylamines. But if still you have any kind of questions related with this topic, so write it into a comment. Inshallah, I will give you answer as soon as possible. So if you like this video, please hit the like button, share this video, and also subscribe the channel to get more interesting video lecture like that. Thank you so much.